Hey everyone, thanks for coming to this week's video lecture. Today we're going to go over chapter 14, where we're going over host defenses and their immunity. And host immunity can be broken down into two different types, innate immunity and adaptive immunity, where innate immunity is something that's present since birth and defined by your genetics, and your adaptive immunity is also defined by your genetics, but also is specific to your immunological memory of past infections. And so it is something that is acquired over time and has a memory of what you've had in the past and helps you fight off similar infections in the future. And so the immune system is arranged in three different layers of defense, where the first line of defense is something that is a barrier that is just in place all of the time to prevent a bacteria from getting to where it is not supposed to be and prevents it from establishing and creating an infection. And we'll talk about how these barriers can either be chemical barriers or they can be physical barriers. Then there's the second line of defense, which is a more cellular response that is generalized to most pathogens that trigger some responsive cells and of the innate immune system. And it also triggers inflammation and phagocytosis and tissue remodeling within the site of infection. We also then have the third line of defense, and this is the area that I study in VDJ recombination, is the adaptive immune system, which is which is a system where you have specific cells that are specialized to specific pathogens. And it does this through a trial and error process. And then it retains those trials essentially to create this repertoire of many different possible responses to all of these many different pathogens, and in essence creates a memory, an immunological memory for those pathogens. So we have the innate immune system, the first line of defense, which is the innate immune system, and it can have it's those barriers that prevent infection that are always active, that are always there. These are physical barriers, these could be chemical barriers, and they can be genetic barriers. Physical barriers are something like your skin is just physically preventing bacteria from getting deeper into your body. You have things like cilia that is physically moving things out of your respiratory tract. You have mucus. Um, Mucus is kind of more of a chemical kind of thing, but I'm just going to go into chemical barriers. So you then have chemical barriers, and these are things that are making it making the environment unfavorable for microbial growth, or for at least pathogenic microbial growth. So think of like salt on the skin and in sweat. You can think of enzymes in your saliva. You can think of acid in your stomach. You can think of that mucus and the enzymes that are in that mucus as chemical and physical barriers. You also have genetic barriers, and these ones are a little bit more obscure, but essentially, for instance, if a pathogen needs a particular protein to enter into your cell, for instance, HIV has a specific receptor that it binds to enter into that cell and infect that cell. If you have a genetic difference within that receptor, you're going to have a different response to such a pathogen. And what we sorry, I have functional movement disorder, and so my brain messes up sometimes. And so what we've actually seen, like clinically, is that there are a few people that have these mutations within this receptor that HIV uses to enter into a cell, and because of that genetic difference, they are more resistant to HIV infections than they otherwise would. And so these are an example of genetic barriers and genetic differences that can create differences in immunity, even at this in such an innate early stage of immunity. You also have the second line of defense, and the second line of defense is things that are more of a cellular response, like I've talked about, where it has inflammation, it has interferons for when you have viral infections. There's phagocytosis that works to specifically consume those bacteria or clean up the mess of the bacteria. And then you have the complement system, which is this big complex that punches a hole in the bacteria's cell wall. You also have the third line of defense, and this is those innate immune systems. These are antibodies. These are T cell receptors. These are B cells and T cells. And these are specifically cells that are specialized to individual types of bacteria. So, sorry, pathogen, antigen, technically. Anyway, excuse me. So I mentioned how the skin is a physical barrier that prevents the pathogens from getting into the muscles. We talked about how sweat changes the local environment of the skin, makes it less favorable for certain pathogens to be able to take hold in those areas. You also have those physical barriers, like mucus and the cilia, that kind of move out those pathogens from the respiratory so that they can't like attach. You also have enzymes and enzymes that work to specifically try to, for instance, break down biofilms or specifically break down specific known problematic proteins. For instance, like lysozymes in your tears. So we also talked about genetic differences already. And so then we have the second line of defense. And the second line of defense is something that's like once the bacteria has broken through, or sorry, I keep just saying bacteria, but it can be viruses as well, or fungus and things like that. But when a pathogen has broken through these physical and chemical barriers, your body has to go through and recognize. They must recognize these pathogens and then respond to those pathogens. Also, just so you know that the virulent factors that are genes like toxins and specific like proteins and genes that make it advantageous for pathogens or make it easier for pathogens to take infections, like are those virulence factors. And those virulence factors are helping us to overcome these first line of defenses, second lines of defenses, and even third lines of defenses. And so, but anyway, once these first lines of defenses fail, the second lines of defenses have to take control. And this is where we kind of get into the field of immunology, where we have specific cell types that survey and circulate throughout the body's tissues and circulatory system and look for material that is not the self. And so they're able to recognize the self and not self, or they're able to recognize those problematic proteins through various systems. And so we talked about how we have that adaptive and the innate immune system and the we have the innate immune system and the adaptive immune system. For the most part, we're going to be talking about the innate immune system in this lecture. But overall, these cells are considered all to be white blood cells, and they circulate throughout the blood and lymphatic system, and circulate and in and around the tissue. And so we have cells that specifically stay in the tissue, and those are resident cells, and then you have circulating cells. And then there are some cells that just kind of move back and forth. And so um, when you have a tissue, you have an extracellular matrix. This extracellular matrix provides support and structure. And a part of that is this reticuloendothelial system, which is a network of fibers and tissues, I mean, fibers and proteins that, and it provides space for these resident immune cells to take up and survey these tissues and monitor them for foreign material. And so, but they also, again, like I said, they circulate in sometimes. And so you can take blood and you can spin it down and you can see the plasma, you can see the white blood cells in the buffy coat and the red blood cells. And these immune cells are in this buffy coat. Just so you know, the plasma has lots of nutrients and chemicals in it, like albumin and things like that. The red blood cells are specifically red blood cells. And just so we're gonna talk about how these cells are differentiated from a specific progenitor cell. And so the those cells that are that are early on in cellular development and they're able to differentiate into many different cell types are called stem cells. And this process of creating different types of blood cells is called hematopoiesis. And we're going to be talking about how these hemopoietic cells are created. And so we have hemopoietic stem cells, and these are our progenitor cells that are occurring in our bone marrow. And then in the bone marrow, this is where these hemopoietic cells are pervasive. They stay there and they grow and they divide and they keep dividing for the entirety of your life. They don't just all turn into these things at the very end. There's like always a supply of these. And so these are what we have in those bone marrows, and then they're surrounded by the tissue of the bone and all of the components of the bone marrow and the signaling molecules that are inside that tissue. And based on those signaling molecules and different fate decisions within the internal signaling and transcriptions within the cell, it will either go into this lymphoid lineage, which is the adaptive lineage. This is where B cells and T cells are developed, where T cells have T cell receptors and B cells have immunoglobulins or antibodies. And you also have natural killer cells, which are specialized T cell. We're not going to talk about very much of these, but otherwise you have this other fate decision where they could go this lymphoid way or this myeloid way. This myeloid lineage makes up a large portion of at least this graph, where you have erythrocytes, megakaryocytes, and monoblasts, which are these precursors to all of these later white blood cells that you're going to talk about, like red blood cells that we talked about earlier. You have platelets, and you have mast cells and neutrophils, basophils, eosinophils. You have monocytes, macrophages, and dendrocytes. These mast cells, macrophages, and dendrocytes, these are these tissue resident cells. These are the cells that stay in the tissue and they just wait for a foreign body to come about. And some of them have specific functions on presenting those cells to the adaptive lineage. Some of them are specifically like signaling, like pervasively, like they primarily signal and recruit cells to their sites, like the mast cells. You also have these circulating cells like neutrophils and basophils and eosinophils. And so when you take a draw of blood and you put it onto a slide and you stain it, you'll see these kinds of cells. You might see a few of these macrophages and stuff, but primarily you'll see these neutrophils and things. And you can tell what kind of white blood cell they are, or what kind of granulocyte they are based on their shape of their nucleus. And honestly, it's really interesting. It seems like there's this process in you have all of your DNA within your nucleus, and there are cohesin rings that like clamp around your DNA, and they like feed it through like a videotape. And uh, it seems like these from this one conference that I listened to, that it seems like um, that process is potentially involved in making these nucleuses have abnormal shapes. Because typically what you think of a nucleus is these round nucleuses, but something about the development of these cells and the signaling of these cells and the like what enzymes are present and what proteins are present within the cell, it causes these nucleuses to be non-round and or lobed and different characteristics. Anyway, again in the weeds. And so we have the neutrophils, which excuse me, I had an itch in my back. You have these neutrophils, which are phagocytotic cells, and these are circulating cells that are recruited into a site of infection after the fact that it's been discovered. You have eosinophils, which are, neutrophils are fairly generalized at attacking things. Eosinophils are primarily considered to be parasitic, attacking parasites. Antiparasitic, they seem to primarily attack parasites. There's not very many of these in the bloodstream comparatively to like neutrophils. And then you have basophils, and basophils are responsible for signaling and helping to further the immune response after we've discovered the infection. You have these B cells and T cells, and these are these adaptive immune cells in the third line of defense. These are the things that after you've developed, like after you've decided that there's an infection, you start to have like neutrophils come in and you start having signaling molecules come in, start being produced, and it kind of creates this like site of infection and this response to it, and it changes the local environment. But some of these cells are antigen presenting cells like macrophages, and their specific job is to, oh, I'm sorry, macrophages or dendritic cells, which one is it? Give me a second. Anyway, one of them is an antigen presenting cell. And it surely says which one it is, and I should know. Give me, truly give me a second. This is funny. What makes it even better is that macrophages and dendritic cells are both antigen presenting cells. So they both have this function. My bad. So what they're doing is they will live in this tissue and they pick up foreign material, or they will phagocytize or swallow up a bacteria, and then they will digest it into tiny pieces. And they will actually take those pieces and put them on their cell surface. And this is so that these adaptive lymphocytes and the adaptive immune cells can come in and look at them and say, I don't know what that is, or I know what that is. That is pathogen X, Y, and Z, and let's kill it right now. And it will trigger that cell to become activated and trigger the immune response. It will actually like proliferate and make more of those cells. A part of that is going up to the lymph node and the lymph system and proliferating, and that's partially why that proliferation is partially why you have swollen lymph nodes when you get sick, is that system. Also, if you want to think about when you get sick and when your lymph nodes get swollen, you start feeling sick and your lymph nodes get swollen. Lymph nodes are a little bit after the fact of after you like have those initial starting to get sick feeling. And so that initial starting to get sick feeling are those second lines of defense, that inflammation that happens, those the tissue damage that might be happening in the response to those things, those second lines of defenses. And then you start having of swollen lymph nodes, and that's the third line of defense. And that's when these B cells and T cells are being activated and proliferating and specifically targeting that specific pathogen that's causing you an issue, comparatively to those first symptoms, which are being just like a generalized response. So anyway, so macrophages and dendritic cells work in the tissue to consume and swallow the pathogens themselves. They work to consume and clean up any debris that's left behind after these cells have been after the infection has taken place, and they also take care of like dysfunctional like your cells in some cases. But that's not what we're talking about. So the other side, we talked about erythroblast and megakaryocytes, and these erythroblasts turns into erythrocytes, which are red blood cells, and then you have the megakaryocytes, which turn into platelets. The red blood cells carry oxygen through the blood using hemoglobin. Interestingly, they denucleate through this process. They take their nucleus and throw it out. This allows them to carry more hemoglobin. If you take biochemistry, you'll understand how the hemoglobin has like a heme group, and that heme group can like associate with different oxygen molecules. And there's some really cool protein dynamics that have to happen, or that can happen, well, that do happen, that facilitate the binding and that for the binding of oxygen to happen in the lungs and the dissociation of oxygen to happen within the tissues. So it's really interesting to see how, like, in detail, how something like that, how something in those systems actually work. And so if you're interested in that, I'm sure there's videos on YouTube to look at if you wanted to not take a biochemistry class and just get to that little piece. Anyway, but also biochemistry is fun, so take a class. Anyway, platelets are these tiny fragments of cells that I don't really understand exactly if they are if they have any roles of like how like sentient they are, how I don't know how many interactions I haven't had very many lectures on these. However, what they do is they respond to wounds and they're helped to create clots and help to establish like the fibrosis that happens at a site of infection and a site of wound healing. And so they can help create layers and help create those barriers to help prevent bacteria or pathogens from furthering getting into certain tissues or into the bloodstream. They can also help prevent like the loss of blood or the loss of like if you have like a rupture or damage to like a blood vessel, they work to mitigate that process. So then we have the lymphatic system, which is what kind of filters and monitors these white blood cells. And so the red blood cells that pump the red blood cells carry oxygen from the lungs to the tissue, and they get pumped through the heart. And the white blood cells are along for that journey, but they also sometimes have to make pit stops along the way. And so you can kind of think of this like if you had like a car raceway, like oh, I don't do like Daytona, like a Daytona race or something. I really hope they have pit stops because that's very necessary for this analogy. And so then you have, sometimes there are certain cars that need to stop for some kind of maintenance, or they need to do something and they take a pit stop. And this is what's kind of happening with these white blood cells, is they are within this circulatory system, within the blood, and they will dissociate into the lymphatic system. And these are lymphatic capillaries and vessels specifically for these white blood cells, and lymphatic fluid, extracellular fluid, sorry, lymphatic fluid, to either monitor what the cells have seen, monitor their health, make sure they're being circulated right. And this happens kind of throughout the body, kind of alongside the circulatory system. You can kind of see the lymphatic vessels here where they span throughout the entirety of the system. And there are there are specific places where lymph nodes or these nodules occur in this lymph system. And these are those places of monitoring. And so the lymph is the liquid lymphatic fluid. What does it? Hey, look, lymphatic fluid. Isn't that what I called it? No, I think I called it something weirder. Anyway, the lymph is this liquid that helps facilitate the movement of these cells so that they're not just like, never mind. And so we talked about this, but because this lymph system goes throughout the entire body, it allows for you to have these cells monitoring all of your tissues, not just like specific things. And so here's a diagram of this system where you have your vessels and your veins, and you have your capillaries where you have your blood coming in and coming out. And sometime in this process, your white blood cells and your lymph will migrate out into the lymphatic system and drain into the lymphatic vessels, and they will collect and go into the lymph node. And this process can happen through sometimes signaling molecules. So for instance, sometimes antigen presenting cells go from the tissue and even come through this lymphatic system into these lymph nodes. And this is where you can think of these lymph nodes as like kind of like a citadel of like thousands of different cells that are just all waiting. And they're all waiting to be like, is there going to be a foreign cell or material that comes by here, and when they do, they all kind of look at it and say, oh, I don't know what that is, and one of those cells is gonna recognize it, hopefully, and then it will go through proliferation. There's also some really interesting structures that happen in the lymph node, and the structures are facilitated by signaling molecules and chemical gradients of those signaling molecules through these tissues. And you can kind of see that that is a central theme with tissue organization and cellular responses, is that chemical gradients are highly important. And it's fairly fundamental because you can think of just like if you have something, it diffuses out fairly naturally. And so you can take advantage of that fairly easily. And so anyway, and so when these cells finally recognize an antigen and they're like, oh, I know what this is, and it's a pathogen, we should take care of this, it will proliferate, and then it will start to specialize and try create, hundreds and thousands of different versions of its, maybe hundreds of different versions or tens of different versions. It kind of depends. I would say tens to hundreds, honestly, of different versions of that to see if maybe one of them is better. And then it can go with that and try to fight that infection. And those cells will then go into circulation, go through the lymphatic system, and then back into the blood, then back into the tissue and the site of infection, right? Okay. So a part of this lymphatic system is the thymus, and this is a place where T cells are developed. And through T cell development. They go from the bone marrow into the thymus, and then the thymus, they finish their development, and then they go into the lymph nodes. And so then you have the lymph nodes we've talked about, and then you also have the spleen, and the spleen is a secondary lymphoid organ, which is a place that filters and circulates and monitors these white blood cells and helps facilitate the monitoring and response to these antigen-presenting cells and different white blood cells. So there's a lot of here. And so we kind of talked about how the first line of defense are these barriers that just already exist. And so, excuse me. So we talked about how these first line of defense like exist and like always exist and are always present and like are just like kind of like walls and traps or like deterrents to the infection. While the second line of defense is these cellular responses. And so these cellular responses take energy and they can't be on all of the time. And they also can be harmful if they're on all of the time. And so you have to have a system to recognize when these systems are needed. And when these systems are needed, it triggers things like inflammation that changes the local environment to make it unfavorable for the pathogen. You have phagocytosis, which helps to consume and eliminate and neutralize those pathogens as well as clean up afterwards. And then you have interferon and the complement system, which are both specialized systems that help fight viruses and bacteria respectively. And so you have inflammation, you have these methods of recognition like the toll-like receptors, which are these family of protein receptors, which all kind of which are all cell surface receptors, and they are like embedded into the plasma membrane of these cells, and they have these like little croppy hooks, and they essentially have this like little binding pocket that can bind and can recognize specific kinds of foreign material. And so these toll-like receptors have a few different kinds. Some specialize in looking at RNA. Some specialize in looking at DNA. Some specialize in looking at specifically proviral proteins, you know. And so these toll-like receptors are specialized at recognizing specific kinds of molecules, but when they recognize these molecules, it can start a cascade of signaling within the cell, and that can cause signaling molecules outside of the cell to be excreted and start this process of foreign material recognition. And it can trigger things like inflammation in those sites of infection. And so you have a pathogen in the tissue and it's trying to take hold and it's trying to proliferate, it's trying to just like live its life. And it has specific conditions that that bacteria wants to live in. And inflammation is a part of a process, a complex process that is very defined. And a part of its process is changing that local environment and making it less favorable for some pathogens. And it also helps to prepare the site ready for the immune response. And so with inflammation, you have redness, you have warmth, you have like temperature increase, you have swelling, you have pain. And these are four characteristics of inflammation. I believe there's a fifth, but I can't remember what it is. But redness is increased circulation and blood flow, increased nutrients, increased sugar, increased oxygen within that tissue. You have increased warmth, you have an increased temperature. And so bacteria have specific growth requirements, and if you increase the temperature, it might make it where it is more less advantageous or harder for pathogen to grow. You also have swelling, you have increased fluid, you have, because of the increased circulation, you also have increased white blood cells that are being recruited into the site of infection. And then you also have debris. It's kind of like a war zone that's happening, and it makes a lot of trash. And so all of that together creates this swelling. And then you have pain. And that pain is as a result of the damage and the signals that are happening around your tissues, which are innervated with nerve endings. So there are a couple of stages to an introduction into a wound. So you have this wound and you have pathogens that have gotten into this wound. You have these specific mast cells that are one of the first couple of cells that are going to respond to tissue damage. They're going to degranulate and release chemicals that signal that there has been tissue damage and trigger the inflammatory response. You're also going to have like dendrocytes and macrophages that are inside this tissue that are going to be looking specifically for these pathogens. And so they are going to be looking for that, and that will be kind of happening simultaneously. You then eventually have these pathogens being recognized and signaling molecules being released into the environment, and they can make their way into the blood vessel. And this can allow us to signal and pull in white blood cells from the circulatory system that are circulating into the tissue, into the resident local site of infection or into the wound. And then the first things that come up are like neutrophils, but then there's things like basophils that help continue this process. But neutrophils are the big workhorse of the whole ordeal. So then, because of these chemical signals, these neutrophils are released from the circulatory into the tissue. This process happens because the actual vessels, the endothelial vessel cells in the vessel respond to these signals and become more leaky and more permeable, increasing the circulation to the area, making more fluid to come to the area. So that's why we get this edema, or this fluid, this swelling. And and slowly but surely, this infection begins to be curbed by the immune system. And then the wound healing process starts to take place, and resident tissue remodeling happens, and the cell debris is picked up by macrophages. So, like I talked about, this process, you have these signaling molecules or chemotactic factors, or signaling factors that are being released into the tissue, and they make their way into a blood vessel. These blood vessels become then a little bit more permeabilized and a little bit easier to leak out just liquids and cells. But then also you have neutrophils that have this process called diapedesis, where there are a couple of different protein receptors that exist on these neutrophils, and they catch on the wall of the endothelial cell, and they kind of they're tumbling through this liquid, this blood. And as they tumble, they'll catch one of those receptors, and it will kind of slow that tumbling down. And then a second one will catch, and then a third one will catch. And eventually it will bring the neutrophil to a halt, or at least slow enough for them to infiltrate through and in between these endothelial cells and start following these chemotactic factors and these molecules that cause these neutrophils to chemotaxis to the site of injury. And so if we have the site of injury that is just pumping out a ton of molecules and signal molecules just through diffusion, if we follow the gradient of those molecules, we'll eventually end up in the site of infection. And that's what's happening, is there's this positive chemotaxis that's happening through these neutrophils to that site of infection to find those pathogens. Another thing that's involved in inflammation is fever. And when you have these bacteria, sometimes they have toxins that they're excreting out, or exotoxins that they're excreting out into the environment. And sometimes those are pyrogens. Other times you'll have molecules within the cell wall that, upon killing and breaking open these bacteria and releasing the contents, you'll have specific molecules that are pyrogens. And these are molecules that, once they get into the circulatory system, they will travel up into the brain, into the hypothalamus, which is what regulates temperature within the brain, and it resets the hypothalamus and increases that temperature, and it causes the body to increase its temperature in response to these molecules. And this is why our body becomes feverish in response to some infections. Um, excuse me. And so we have those exogenous pyrogens, which are existent inside those cell walls and are produced and released into by the bacteria itself. LPS is not released by itself. LPS is only released after the uh degradation of the bacteria, I believe, or like after the killing of the bacteria. So I'm not really sure why it's being called an endogenic pyrogen. But then there's endogenic pyrogens, which are molecules that are being released and caused through that like elimination process, right? So we have excreted agents, and then we have like subsequent damage that's happening because we're trying to eliminate them, and like the trash is essentially causing us to go into this feverish state. But again, this feverish state is an increased body temperature. And if you think about the conditions needed for certain bacteria and pathogens, potentially that higher temperature is less favorable for bacterial growth, for that pathogen's growth specifically. And so we kind of talked about that a little bit. Also, there are subsequent interactions, like for instance, that increase of temperature impedes the amount of iron that can be reduced by that bacteria. And so it kind of takes away some metabolic advantages some pathogens can have. We also have this phagocytotic process of searching and finding these pathogens and eliminating them by engulfing them and taking them into our cells and then turning them into essentially rubble. You have neutrophils that are doing this, and you have macrophages. Neutrophils are general purpose, and they are circulating through the circulatory system and then come out into the tissue when they're needed. And then you have macrophages, and those macrophages are resident in the tissue, and search for debris and foreign material, and then they'll bring that in and present that to a T cell for evaluation and triggering that like third line of defense. You then have eosinophils and dendritic cells, which both also work and can conduct this phagocytotic process of identifying these signals through a receptor, and that receptor signaling a change in the morphology of the membrane. And that change of morphology of the membrane causes this to create this like this gulf or this crater, and that crater can have contents, and that contents hopefully is bacteria. Sometimes you can have bacteria that are covered in what are known as PAMPs, or proteins that like recognize specific patterns on these bacteria, and it can coat these bacteria so then they be recognized and stuck to those receptors, and then those receptors are engulfed into the bacteria and degraded through an endocytosis, an endosome process, endosome digestion process, Lord. But also you can have this kind of engulfing process be a little bit more random, where we're engulfing just the extracellular matrix and what is in the environment, and hope that there is that bacteria there. Obviously, there would be something that would like trigger that thing. But we can see here where we have a phagocytotic cell where we have the pathogens or the bacteria on the cell, and the cell trying to engulf those things. Anyway, so when you engulf these bacteria, they turn into a vesicle, just kind of like a little bubble that has been internalized into the cell. And then that vesicle slowly becomes acidified, and acid and proteins are being secreted into these vesicles and turn them into endosomes. And these endosomes break down the cellular components into these tiny things. Some things might be able to be utilized again, but otherwise it's then just released back into the environment and they go on their merry way. Interestingly, there are some times where these phagocytotic cells or macrophages can like gang up and like merge together to like try to engulf something really big, which is really fun. I still don't really understand that exact process, but they like literally merge together and become multinucleated. So it's kind of like a supercell or something. That's not a real thing. I'm just making up that term, but like feels like that, right? So anyway, you also have these generalized, so what we've been talking about are these generalized processes that kind of are the response to any kind of pathogen. And this is why like inflammation, joint pains, and redness and stiffness and like all of those things are because those are generalized symptoms that you can experience because these responses are generalized, regardless of what you're sick with. And so sometimes you can be sick with different pathogens and have those same symptoms. That's why like you can have like rhinoviruses, you could have a cold, and you can have a respiratory virus, or you can have like bacteria in your lungs and like an infection that way. And like sometimes their symptoms overlap. Or for instance, it's because they're utilizing those same response and some cellular signals. Additionally, part of the reason you have some of those same responses to vaccines is because it's triggering those same immune pathways. And so subsequently, you get the same symptoms because they're activating those same they're activating those same um, pathways. Anyway, while those are generalized and kind of work towards uh, the ultimate end goal of any pathogen, there are also specific systems that work towards eliminating specific species or specific kinds of pathogens. So for instance, interferon is a signaling molecule that works to inhibit proviral transcription and viral infections. And then we have a complement system, which is this large complex of proteins, which assembles on to the surface of a bacteria and literally punches a hole through the plasma membrane of the bacteria and makes all of like its cytoplasm come out and essentially makes it like deflates it like a balloon and kills it. And so this system is specifically designed for killing and neutralizing bacteria. And so with that, that is the last of chapter 14, and I hope that you learned something awesome.